now share the screen. All right. So I tried to make the edits that the font is bigger on these slides. So those that are having trouble reading those, I made the effort. So we're chapter 10. So we're moving it still into what is known, if you look at the like the table of contents, we're in the immunologic procedure section. So we've done 9 and 11, now we're backing back into 10, and then we have 12 and 13 to go through. In lab today, we have continuation of the ELISA. This time we're switching it where we're actually going to detect the antigen instead of the antibody. We went for the antibody first, so the technique is basically identical, except you're setting it up a different way. So you'll go through it just like you did last time. So it gives you another run of the ELISA, and we'll see if those that got a little semi-positives in some of the wells, this time we hope we clean that up and we get the blue where it needs to be and the clear where it needs to be, so we have some great technique today. So if you were a little, I won't say sloppy last week, I mean on Monday, be more careful today. I think we gave you some troubleshoot ideas about paper towels, bubbles, anything that could bleed over or come across and get in that well somehow, some way it did. That's okay, we'll do it again. This chapter, we're going to hear about another lab we're about to take on, which is immunodiffusion. Okay, so we do electrophoresis immunodiffusion, where it's another, you're going to actually pour gels. So we're going to have to work to get, it's good that we're smaller groups in these, and I want to make sure that we can each pour a gel and run the gels. I think we can do 3. I think that's the way it works out. I want to say we did 2 last year, but I can't remember exactly yet. We'll get into it and make sure. But that's on the deal where we will actually work on prepping, pouring gels, running it, and hopefully you'll get to see that result. But we're going to talk about that today in chapter 10. So we're trying to connect what we're talking about to what we're doing, trying to do the best we can on that, so that it makes sense. Once we talk about it, you go down and you actually perform the actual test. So we're going to try to keep that going. So we're going to move that electrophoresis up from last year and put it right here with after chapter 10. Questions? All right, so here's what we got to do. We're going to talk about antigen and antibody binding. We're going to talk about precipitation curves, light scattering techniques, passive immunodiffusion, electrophoretic, agglutination, instrumentation, and finally quality control. So it's not a whole lot of immunology now. So I hope you're seeing that. This kind of lightens up the difficulty, I'll say, in the information. This is more like, hey, this is why something's going to work. These are some definitions you need to know. Now let's go do the work or learn the technique or understand the technique. So this is always what I call, this is the CEO comes into the lab and asks you a question. They don't know anything except how to run the hospital. So what you're doing in the lab, they don't have a clue. So unless they were a lab tech, which we don't have very many of those, but they might ask you, like, even when you're at a clinical, as a clinical student, they may come and ask you how that works. Okay, how does that machine work? What am I spending my money on? Like, and you're the one that needs to answer that. So what you're learning is like how these tests work. Okay, keep that in mind, because that's an uneasy feeling when your boss comes in and says, "Tell me how this works. I just spent $200,000 on this machine you're working with. How does it work?" "I don't know. Tell me." And you go, you hit this button, right? That's not the answer we're looking for, right?
Is there any way before clinicals to know what machines they have where you're going?
Sort of, kind of, yeah. I mean, it really, yes.
So you can like look over what.
Yeah, look over maybe what the good thing is they need to orientate you once you get there. So I would wait on that. You're going to do modules like for chemistry. That's one that always pops up. Like, what chemistry analyzer do you have? What modules did I do? The techniques are all kind of there. The running of the analyzer is pretty much the same. But I like if you go in and they get to orientate you on their analyzer. They'll spend time, hopefully, doing that. They're supposed to.
Well, in clinical chemistry, we did the Siemens modules like on the computer or whatever, but I don't learn anything from that. I don't, zero.
Right, that's why I'm saying you might as well just wait till you walk in,
Okay.
so they can orientate you on it. Yeah. That way, you didn't get it out of the module because you weren't getting to put your hands on it. So now you're going to get to walk in, and they're going to let you stand in front of it and do the actual operate. That's what I would do if I was your clinical precept. So, yeah. And that's what, when we say that, oh, there's, I did this for NACLs was I used to put all the analyzer names that everybody had. And they're like, just put chemistry analyzer, just put hematology analyzer. Because baseline for accreditation is we give you an experience. And if you say, well, this hospital has this toy, and maybe the student feels like, well, they didn't get to go there to play with that toy, but they still had a toy to play with. So you just say you got a chemistry toy to play with, you got a blood bank toy to play with. So we keep it real baseline, but they all basically operate the same. I mean, you're gonna learn that. Some you'll like. And what we usually find is the student that trains on certain instruments will either have a good impression, and once they get into a position to buy that analyzer for a lab later in life, or they might not. We all have our favorites, and I'm really big on favorites. I have certain analyzers I like. And it's just because I've gotten real familiar with it. I know how to troubleshoot them and all that good stuff. Hope that answers your question.
Yeah.
Okay. All right. Antigen-antibody, and let me try that again. Maybe I should say antibody-antigen binding, right? And we have a word known as what? Affinity. Affinity versus avidity. What is that, right? Affinity is the initial attraction. So it's what draws the two together. Okay, so that can be really strong and be really weak. Doesn't mean the binding is going to be really strong and really weak. It just means that first attraction to come together is known as affinity. Okay, so it's between the Fab site, right? In between, Fab, and of the antibody and the epitope or the determinant site of the antigen. So that's where this part of the, where I'm going to link to the antigen, that's the attraction. Okay, so if the epitope is really strong, it's really antigenic, then we have a strong, affinity, right? Because there's a strong attraction to that initial bonding. That's the attraction force. The strength of the attraction, I just said specificity of the antibody for a particular antigen. So we know that. You've already learned this. This is reinforcing that idea. So here is the affinity. The more the cross-reacting the antigen resembles the original antigen, the stronger the bond will be between them. So here is the antigen, right? Higher affinity, okay? Meaning that's going to be like that one. There's more, likeness that I want between that antigen and the antibody. As you see, you got positive charges, that's got a negative. And you see here that's lower affinity. So that's not as strong right here because that antigen has some, but not the other cross-reactivity. So we see that with certain antigens, we have cross-reactivity. Like we say, oh, there's a specific antibody formed for each antigen. Well, some antigens have similarities between the two, right? And we're seeing that hopefully with COVID. And, we're heating up a hot spot here in this region, so it's stay strong. But yeah, we got labeled yesterday as the highest trending area of the state yesterday.
The highest?
The highest. We got a 9% positivity rate for people getting tested in this area right now. So keep that. The school's numbers look great. Okay, the ones we post every day on the school website look good, down to 50 total. But the area looks really red on the map.
Didn't MacArthur get shut down now?
Who's that?
MacArthur?
Yeah, they all.
My son updates me with that every day. Yeah, I've heard the junior high. There's a lot of mess with public schools right now, but we're going to stay strong here and be here every day. And my son will keep me updated on how many times he feels like, well, their school's shutting down. Why can't we shut down? So, yeah, they're just waiting on that day. They go, we're shutting down, just like that. Okay. Little Rock's another story. It's a mess. Those are from down there. They're having virtual strike today. All the students are striking on their virtual education. They're not going to log in today on purpose.
What?
Yes.
Why?
Wow.
Because they want to go to school?
They're high schoolers. I don't know. They're supporting their teachers who took a day off last week. So yeah, it's a mess. All right. So we're trying to stay with a plan. We got a plan, and we're staying with it. And the whole idea is to get through the plan so that next semester doesn't look anything like this semester. That's all right. That's the hope. All right. So now we got avidity. Avidity is different than affinity. Avidity is the sum attraction forces that happen when we lock up. Okay. So there's attraction with affinity. Avidity is once that is there. So how strong is this? Okay, once the antigen antibody gets there. So it's the strength in which multivalent antibody binds to multivalent antigen, and it is the overall stability of the complex. Okay, so strong avidity means it's going to be harder to get that broken away. Okay, still happen, but it's going to be a little harder. So that's the main two things to think about today when we think about antigen antibody. So then your book goes into another state. It says that, hey, we got the law of mass action. It's a way of having a K constant value figured. Okay, so this would be a measurement we could do. And this is the law of mass action, where we take the antigen antibody concentration. So this is the complex, okay, together. That's the antigen AB right here on top. And we divide it by the free antibody and the free antigen. The concentration of those. So if we had a mixture, all right, we put those two together, then we can do a K constant and we can say, okay, the K depends on the strength of the binding, because if it's not a very strong bond, then we probably got a higher concentration of free antigen and free antibody when we do this measurement. That makes sense? So if it was a real strong connection, then we shouldn't have as much free hanging around in the concentration. So if it's strong, what happens? The K constant is going to be bigger because this number is bigger and it's on top. And we know from math that if these are bigger, then we have a lower K constant because we would divide out and get smaller. So the larger the amount of antigen antibody complex, the higher the K value. So that's a test question. Test question says, if you have a high K, what does that mean? That means you have a larger amount of the complex antigen antibody together. Okay, and that would mean we had a good strength, a strong binding between those two. And if you have a higher K, we know with agglutination that that's more visible, easily to detect, because you've got a bigger complex that you can see with your eye. Okay, so when we have the plasma running around and the red cell running around in a test tube, if they're not locking up together, then it's harder to see, right? We know that from our 4+ versus our 1+ agglutination reaction. When we do precipitation, these are the kind of we're going to have to review the reactions that we got going on. This one's a fun one because this has some really cool test questions with prozone and postzone, and you'll need to know who's who, like who's where. So if you look on your book on page 142, I'm sure I got a picture on your slides. Pretty sure it's coming. Yes, there's a slide picture that shows this, but the whole idea is the equivalence versus prozone versus postzone. So we're going to, with precipitation, right, when we precipitate something out, we combine a soluble antigen with a soluble antibody to produce an insoluble complex that are visible, okay? So it requires that the antigen antibody have multiple binding sites, equal relative concentration of each is what we want. So when we precipitate out, what we're saying is that if it binds up, right, becomes visible, we can see it, becomes insoluble, so all the other stuff would be soluble, right? So how does this work with the prozone? So here's what I was talking about. This is the antigen antibody complexes on the y-axis and the antigen concentration alone on the x-axis, okay? So as you see, the number of multivalent sites, the antigen and antibody are approximately equal. That is the zone of equivalence. So we get this right here. That's the big complexes that form. There's no extra. Everybody links up and we're all happy, right? So we get this bell-shaped curve, right? That gives us, as the antigen concentration goes and the antibody complexes forms, right? That if it's equal amount of both, then we have a zone of equivalence. So to be detectable, we have to have a precipitation reaction must be run in the zone of equivalence. So we need to see this to detect it. The prozone, remember over here, this is where if you look at prozone, we have excess antibody. So that's the key. To a prozone phenomenon, we have antibody excess. So that's the way the test question is going to roll. They're going to say, oh, you have a reaction and you see this, and would that be prozone or postzone? And basically what you're going to say is, if I can tell there's antibody excess, then we're going to go prozone, in front of the zone of equivalence. The antigen combines with only one or two antibodies. There's no cross-linking, there's no lattice form, and what is really important is that this is where false negative reactions occur. Because they occur because there's so much antibody there. If a false negative reaction is suspected, we need to dilute out the antibody and perform the test again. It may give us a positive result. Okay, so we just can't pick up that there's not enough antigen antibody together at the same concentration to form a positive test. So if we're looking at a agglutination or precipitation reaction, you know, we're going to judge that. You think about agglutination with staph, right? Coagulation, right? We did a coagulation, put it on there, and we mixed it, and it clumped up, right? But if we had loaded that up with too much antibody, right? And there wasn't enough antigen in the solution, there wasn't enough staph in the, say the antigen wasn't there with the staph, then we wouldn't get the strong agglutination. We may miss it, okay? So we might need to dilute down our antibody. So there's a kind of a fine line how we test with this idea. But this is kind of a false negative connects with the prozone phenomenon with antibody excess. So you want to keep those three things together. The postzone phenomenon would be right opposite. We got a little bit of antibody there. We got more antigen there. So there's small aggregates surrounded by excess antigen, no lattice is formed. So neither one of these is going to give us what we're looking for. The presence of small amount of antibody may be obscured, causing false negative results again. How about that? The test is repeated about a week later with a specimen to give time for further production of the antibody. If the test is negative again, it's unlikely that the patient has the antibody. So this is what we're looking at right now with COVID. Okay. COVID is, do you have enough antibody to test? Do you produce enough, right? Do you went and gave blood, you didn't test positive for the antibody, but does that say that you had it or not had it, right? don't know, right? Maybe you didn't like, so this helped me, right? This idea of, okay, I'm gonna give myself a week after I test positive, to make sure that I gave myself long enough time to actually, once I was over the virus timeline, give myself a little more time to make sure the antibody is produced and make sure that the test is going to be correct. So we have patients that run right out, right? They want to know, I want an antibody test. Well, we knew there's, have you been exposed? I don't know. I need an antibody test. I think I was. Somebody told me I was with them like yesterday and they're positive today and I want to get an antibody test, and that's not really what we want to do. So at the beginning of this outbreak, we had a lot of confusion and definitions that had to be explained, like the press conferences were basically to explain why we're doing certain tests and what that means and why you need this one versus that one. And the timeline was, it's time to test for the antigen. It's still time to test for the antigen, okay? Still active cases. There's still people getting it. So we're still looking at who has it, right? Maybe when the vaccine comes out, maybe it would be a good idea to start heading toward the antibody line of the test to say, well, if you hadn't gotten exposed yet, the vaccine would be great for you because it would give you protection if a second wave comes through, or you have somebody, travel in, or you went and played somebody in a sporting event down in Texas versus New York, or wherever you may be going. That might be the time to do the antibody test. We don't know, all right? We think that's the timeline we want to look at. But again, both the prozone and the postzone can cause false negative results. But it's the difference in which one's in excess. So you have to determine on the test, when I say we have antigen excess in the test, is that a postzone or a prozone? Got postzone, okay? It's not overly hard. I don't think it's too complicated, but it has been confusing to some before you. Precipitation reactions, here they are. We have nephrometry, where it's a sensitivity of 1 to 10, so it's pretty sensitive. And you pick up 1 to 10 micrograms of the antibody per milliliter. This is a light scattering technique, and we don't have a slide picture, but you see it here at your page 143. It shows you that you have a cuvette, the light source runs through. It's kind of like a spectrophotometry, but it's a different it scatters it instead of just absorbing it. So the more complexes you have in your system, the more light gets scattered, and it detects that scatter. And thus it measures it as more scattered light means more antigen complexes, okay? Radio immunodiffusion. This is where we're going to check for immunoglobulins, or complement can be done this way. It's not as sensitive, as you can see. It picks up 10 microgram levels of antibody per mil to 50. And this is where the antigen diffuses out of the gel. So this is kind of what we're kind of doing with our test when we do immunodiffusion. We're going to take an antigen, we're going to put it in the gel, and then we're going to have, the gel is going to be infused with antibody, okay, and we're going to see movement of the antigen antibody toward each other. Okay, so we'll see this precipitation arc form in the gel, and that'll help us determine the amount of antibody. We have Australoni double diffusion, and this is always fun because this one shows up. Every board question I've ever seen always has the double diffusion thing show up. What I mean by this, right? This is Australoner's patterns. We're going to cover those in a minute, but that's a picture that's going to show up on a test. You'll get one of those and they'll ask you what's going on and what is it. So don't lose sight of just because it sounds like an old way of doing things. Australoni double diffusion detects complex antigens such as fungal antigens. Not as sensitive, 20 to 200 microgram antibody, so a little more has to be there before it's detected. Both the antigen and the antibody diffuse out of the wells in the gel. So we'll place those in there and we'll get another thing known as line of precipitate. And the way that sets up will tell us what we actually have. Okay, and we'll go over that in just a minute. Immunoelectrophoresis, that's what we're calling what we're going to be doing, but it's not quite like this. We're going to differentiate serum proteins. This is more of what a real immunoelectrophoresis technique does, because then it gives you a level of the different antibodies. Okay, so electrophoresis of the serum followed by a diffusion of the antibody from the wells. So we do a little bit of that. We're going to run an electric current through the gel. We're going to see a migration, and then we have wells set up with the antigen, and we're going to see those move. So we're kind of going to mix these techniques in lab. Immunofixation electrophoresis. This is variable with sensitivity, but it's a direct application of the antibody to the gel. And this is the one that's the classic. Looks like this. Okay, so you can see that you get a gamma line, there's no alpha, there's a mu, there's a kappa and a lambda, and you see these show up at certain points. This is kind of like your control line. So this is definitely, you picked up an IgG and a lambda. Does that make sense? I mean, I think y'all seen this probably before in a technique, a gel, Western blot type deal.
Is this the big like gel slab that's got the slots in it? You can pop it.
Mm-hmm. Yeah, we're going to do that. Yeah, we're going to use that same instrumentation that you use in chemistry for a different protein electrophoresis, and we're going to use it for immunofixation.
But it works the same way as that.
A little, pretty much. Yeah,
Okay.
we're gonna go over that when we do the lab, though. All right. So let's look at these. This is radio immunodiffusion. So we're going to pick up, we're basically, where are we? We're back, go back one, right? We kind of had them all together. This is on 143. A single diffusion technique. The antibody is in the support gel. So we're going to load the antibody into the gel. The antigen is applied into a well. So you cut the gel. Once you form the gel, you make a well, and it's, let's just say it's not just overly clear on how to make that, but we made it. We stuck something, I can't remember what we had, but it didn't look like it would do what it did, but it did. It made it cut, and we pulled out that, and then we loaded, and it was real important because whoever had their hands on the pipetter just unloaded it and missed the well. So on that gel, we had this big smear of, it still kind of worked, but we weren't being real careful when we followed directions on getting the tip down toward that cutout and loading it slowly into the cutout so that it stayed right there before we started. Just a note from last year. The antigen diffuses out until the point of equivalence is reached. So there's an endpoint method, and then the square of the diameter is proportional to the antigen concentration. It's just one of our immunodiffusion techniques. This is it here, and this makes sense because this is the cutout, right? So there's a glass slide coated with agar containing a specific antibody, and you see the more that it diffuses away, right, out into the gel, then that's the higher concentration, right? So the concentration is more. These are standards, and here's our unknown, and we compare our unknown to our standard line. So this is how we read a standard curve. So this is a straight line for standards, right? So that's how we measure. You hopefully had this in principles, but I'm not so confident you did. Determine the precipitation ring. So this is basically how we measure a lot of stuff in lab. This is great, right? This is a standard curve. So we have 3 known values, right? And we measure those, right? So we have those. And then basically the analyzer or you just compare the result of the patient to the standards and come up with a value. So if this standard was like 6, and that one looks like it's like 16, and that one looks like it's like 26, okay, milligrams per deciliter of the reactants, then our patient ended up at about, what, 11 or 12? Okay, so we compare known standards measured versus the patient. The patient gets put on the standard curve plot. So we plot that. So if you like doing graphs and things and doing graph paper, making these standard curves. And then there's rules about them, you know, the straight line that connects the most dots and that kind of stuff. But that's how we do it. This is clinical chemistry. I mean, that's how the analyzer works, measures, but we also do it in immunology to measure the amount of antibody, okay? Or the amount of antigen. Depending on how we
on a picture, because I remember taking a picture of it, so we had to load it or load it to the blackboard or whatever.
Okay.
And submit it. That was a long time.
Australoni diffusion is a double diffusion technique. The wells are cut in the gel, and both the antigen and the antibody diffuse out radially. So we have an antibody in one well, we have antigen in the other well, and a line of precipitation forms where the antigen and antibody meet. So what is driving this? What is pulling these together? What is the attractant force again? What is it known as? Affinity, right? Not avidity. Affinity brings them together. So affinity, that's that attractive force. So through the gel, we can bring these, they'll migrate. They move through the gel toward each other until they form that line of precipitation where they form the complex together. There's three possible IDs. There is identity, there's partial identity, and there's non-identity. So those are the three. And this is what we're saying. These will show up. You'll see these on a test, okay? The picture will be there. It's in the board review book. It's in my test. It'll be, and it just shows a pattern of diffusion and says, what do we have? And what you see with identity is a curve that is not cross-linked. It doesn't cross over like a TP. Okay, it's just a mountain look to it. So here's the antibody. Here are the antigens. Okay. So they're the same antigen here. So you would think that yes, the antibody would migrate equally out to both of those at the same rate. It would end up being equal line. It makes a nice arc there for identity. A non-identity, okay, here, non-identity would be where we have a crossover. So the TP look here. Okay, so here are our two antigen samples, and there's no identity to either one. It didn't come to either conclusion, so there's a non-identity if it crosses like a TP, okay? Here's partial identity, right? And the partial identity says that yes, we had the non-identity link for one, but the antibody did like number 3. So these are different antigens in those wells. Okay, this would show you yes, both were the same. These were two different and it didn't like either one of them. Okay, so reacts with 1 and 3 is a dotted line, right? Reacts with 1. So here's reacts with 1, reacts with 2, okay. Now, somebody want to help me with that antibody key? What's that set up like? What do you think? What does it acts with 1, acts with 2? Acts with 1 and 3. Let's look at it. From your text. See, everybody got their text? Anybody want to help me? Zoomland, you want to help me with this one? Let's take a look at it. An antibody mixture is placed in the center well. Unknown antigens are placed in the outside well. The antibodies and antigens are all diffused radially out of the wells. In A, the serological identity, if the antigens are identical, they will react with the same antibody and the precipitate line forms a continuous arc. So take that line. What did we say? That if the antigens are equal, right, the same, then we get an arc of identity, correct? with this antibody. All right, so here comes the confusing part. B, non-identity, if the antigens share no identical determinants. Okay? So is the antibody mixture the same? So you put this antibody in, they don't share. So what are we really identifying here? Differences in the antigen, right? Seeing it or not seeing it? Seeing that part? Got one saying no, one saying yes.
So they're two different antigens, right?
Two different antigens, right? So the antibody it will react with who? So are we determining here, like this, here's these are the same antigen, right? And this antibody is reacting with one. So the antibody reacts with the first antigen, okay? Here we just had the wells of both antigens were there. So if we were testing a patient and another patient, right? We're like, are y'all both sick? You both have the flu, right? And we put their antigen in the wells and we determine that the antibody that we have here against that antigen is reacting with both patients. We call that serological identity, but we're just saying that these antigens are the same, okay? Here, the non-identity, right? What are we saying? The non-identity, if the antigen shared no identical determinants, they will react with different antibodies and two cross lines are formed. So remember what we put in that first well. In the first well, we put an antibody mixture, okay? So this is a mixture of antibody here showing that this antibody mixture is reacting with who? Does it react with two? Does it react with one? Yeah, I mean, it's moving, right? It's moving out to both of them.
What if it wasn't a mixture though? Wouldn't it just? It would just react to whatever antigen.
Let's go back. Agglutination diffusion is a double diffusion technique. The line of precipitate forms where the antigen and antibody meet. Let's look. Y'all with me?
So we don't have a mixture of antibody in A, right?
Yeah, I mean, the mixture is this.
Okay.
We're testing for antigens, right? Let's read the technique again. We got time. The Agglutination double diffusion is a technique, all right? Immunochemical technique. It is the technique both antigen and antibody diffuse independently through a semi-solid media with two dimensions, horizontal and vertical. The wells are cut in the gel and reactants are added to the wells. Most plates are set up with central wells surrounded by 4 to 6 equal distance outer wells. Antibody that is multi-specific is placed. Okay, so multi-specific antibody would mean a mixture that would detect many different antigens. Okay, that's the first key to the technique is we're not just putting a single antibody in the well. Okay, what we're trying to determine is the antigens the same or different, or what is the similarity between the antigens? Okay, that's the first step. All right, so antibody multi-specific is placed in the center well. Different antigens are placed in the surrounding wells to determine if the antigens share identical epitopes. Okay, so what we're doing is we're testing the antigen out. We could put this in for coronavirus. Let's put coronavirus COVID-19 versus coronavirus 02, COVID-02. Let's put those in these wells and just see how similar they are, right? So the similarity would be tested. We could do that. Diffusion takes place radially after incubation period of 12 to 48 hours. The precipitin lines form where the moving front of the antigen meets the antibody. The point of equivalence is reached. The density of the lines reacts to the amount of immune complex formed. The position of that precipitin band, right, we got 3, between the wells allows the antigens to be compared to one another. Several patterns are possible. The fusion of the lines at their junction to form and represent serological identity. A pattern number 2 is cross lines demonstrate 2 separate reactions and indicates the compared antigens share no common epitopes. That's number B. Or there may be fusion of 2 lines that spur, so we get the little spur up there on number C, indicates a partial identity. In the last case, the 2 antigens share common, but some antibody molecules are not captured by those antigens and travel through the initial precipitin line to combine with additional epitopes found in the complex antigen. Okay, so that's where we need to keep it straight. These are to test what? The antigens, okay? So this serological identity, there's reaction. If they're identical, we don't get a spur, we don't get a cross, we get a smooth arc for identical antigens. Epitopes are the same, found it, they're similar, they're the same, they're identical, right? So if you want to say identity is identical, that's a good way to think about it. And then non-identical would be that one antigen versus, or just say the antigens are different, okay? They don't share the epitope, so we get the cross. And then partial, meaning we're not identical, we're not non-identical, we're partially identical, which means there is similarity between 1 and 3. There are similar epitopes, but not identical. So you get a spur. How's that? Y'all clear as mud on that one, or you feel better? Hopefully. Okay. Hopefully you do. All right. Precipitation, immune fixation, electrophoresis. It's a double diffusion technique, just like Australoni's. Unknown antigen is electrophoresed, and then the antibody is applied directly to the gel. So we take our antigen sample, our unknown antigen, we place it in the gel, we electrophorese it across the gel, then we apply the antibody directly to the gel, and it precipitates form where antigen-antibody combination takes place in the gel. So this is kind of, we're going to see this right here when we do it downstairs. It's used with serum as the antigen to determine over and under production of the antibody types. Ours is not going to look quite like that, but we will see some dye go across. But what we're going to look for is a precipitation line, so it's a little different. All right, and then we'll spend the rest of the time on. So you're going to get to do that. You're going to get to do kind of a mixture of those diffusion, electrophoretic, antigen-antibody applications, running electric current through the gel, and we'll get to see that precipitin line form. All right, so this next part is key to blood bank, okay? Agglutination reaction. Anybody taken blood bank yet?
I don't think you can.
I was just checking. I was just kind of double checking everybody, make sure that you had to get through this class first. Yes, that is the progression. Agglutination is a visible aggregation. We can see it with our eye. We don't need any more. We don't need any fancy machinery to pick it up. We just need our eyes and a light, maybe a magnifier too, agglutination lamp. So it's a 2-step process. So a 2-step, first is their sensitization. Can I not say that either? Sensitization, good, I got it. Initial binding, the antigen antibody unite through antigenic determinant sites, and then there's the big lattice formation. So we kind of sensitize it, get it kind of primed up, and then the lattice forms to form a large aggregate. Okay, so this would be maybe an incubation period. right? We put the two together and kind of let them incubate together, and then we spin them down. Once we sensitize the reaction, then we spin them down to clump them all up together and get the lattice formation. So that's our agglutination, and we can, it's with antibodies, they're called agglutins. So this is it. We got our antibodies on the left, we got our antigens with multiple antigenic determinants, and we put them together, and we see no visible reaction at the beginning, but we're getting there in the incubation time. We're kind of letting them get to know each other, and then we'll do a spin down, and we see the lattice formation, a clump in the bottom of the tube, and we shake that apart. It makes a 4+ pellet, and we're like, we got agglutination. We feel confident that we have a positive reaction. Okay, so we'll do this with blood bank. This is your red blood cell here, and we put blood bank in the middle of it, and these are your antibodies against the red blood cells. So if this is an A red blood cell, this is an antibody to A, type A blood, and we put it together, and you see that big lattice formation. I think you've seen this already. Shouldn't be new to you here. So there are types of agglutination. There's direct, and it uses known bacterial antigens to test the presence of unknown antibodies in a patient. One that we know is Weidel test. If you're in micro, sorry, Preston. And we've already gone over the Weidel test, right? You had her on a test. Rapid screening test to determine the possibility of typhoid fever. No, we hadn't had a test on this yet, have we? All right, so this is a good review. Use the Salmonella O, the somatic antigen, and the H antigen, which is the flagellar. And we can do a Weidel test to type out Salmonella. And that's where we use a bacterial antigen from the Salmonella. That's the somatic antigen versus the flagellar. And we see if we have unknown antibodies produced against those. So we would test ourselves for Salmonella. This is direct agglutination with hemagglutination with the red blood cells. This is our red blood cell grouping, and we're going to do this all next semester in blood bank, but here we are, right? And this was what we saw with our negative that we were hoping for our immunodiffusion, our antibody diffusion titer, and this is what it looks like when it agglutinates. It comes off the side of the pellet, breaks up. So in our grading scale, let's call number that one what? Is that 1, 2, 3, or 4? It's positive, so this is the one we're judging. So let's all say what we're going to call that. Is that 1, 2, 3, or 4? Who says 4? 4+. Okay. Who says 3? Who says 2? I'm going to go 2. I probably will fall asleep. And then who says 1? Nobody says 1. So but does it matter? Yes, in blood bank it's going to matter. Okay, so you need to start practicing. We'll say, oh, positive, positive, just call it 1 something, but in blood bank, it's really important that you know how to grade these because the strength of reaction can make the determinant of how many antibodies you actually have. If you only got 1 versus 2, right? Or you got what we call single double dose, but Dr. Walls is going to cover that. But just think about if we got an antibody, that unknown antibody in the patient's serum against the red blood cell antigen, not AB, because we're smart enough not to do that. But what if they have a KEL or a Duffy? Okay, those are the antigens you'll learn next semester. So it's very important that you come up with your grading scale and you make that determinant in the tubes and the gels on what you're going to call agglutination. So here it is. We got a red blood cell centrifuged here. We get a 4+, 3+, 2+, 1, barely. And then you got little descriptions, large clumps, numerous, small. That's why we went with 2. Barely can tell, that's 1. And then smooth out, nothing there. That was that other 2. We have B, which is a, what is this? That's solid phase, I think. Is that what we're doing? Button formation. I think that's what they call it on B. Rapid slide method, strong agglutination, okay, big clumps, smaller clumps, negative clumps. This is what we do in immunology. We do a lot of this slide test, right? That's what we're going to be doing. We're going to do a lot of agglutination testing on slides, and you'll be judging them on the slide in front of you. There's passive agglutination. which employs particles are coated with antigens not normally found on their surface. Erythrocytes, we can coat them with latex or gelatin or silicates. Synthetic beads, we can use that for agglutination. And that means we can detect rheumatoid factor. We have that test kit downstairs. We do group A strep. We have that test kit downstairs. We don't have viruses for rotavirus or cytomeglia or chickenpox. Hepatitis viruses and HIV, they're all used with passive agglutination. Process is the antigens attached to a carrier particle, and then we're looking for the antibodies produced by the patient. So if we get agglutination, it's a positive test. Reverse passive agglutination, used to detect microbial antibodies. Common applications with our group B strep, our Staph aureus, groups A and B, strep group A and B, rotaviruses, cryptoneoformans. The process is the antibodies attached to a carrier particle, so it's reverse. So instead of passive where the antigen is attached to a carrier particle, in reverse passive agglutination, we attach the antibody to the carrier particle, and that's the key difference. It's real easy. Still get agglutination if the sample is positive. So you have pictures of that showing that. Here is the carrier with the antigen. This is the carrier with the antibody. So this would be passive agglutination. This is reverse passive agglutination. All right, and lastly, we want to talk about is types of agglutination reactions. And this is one that we will spend a little more time on because it is based on competition. Okay, so what happens is we use agglutination inhibition to detect antibodies to certain viruses. Because what we're going to see is that the lack of agglutination is a positive reaction. And that's just switching gears, and that's why we save it for last. That's why we talk about it last. Okay, and what that is we use it to detect rubella, mumps, measles, influenza, adenoviruses, respiratory syncytial virus, herpes virus. So what happens is, finish up, it's the last thing we're going to talk about, so hang in there. We have no antigen, right? Patient has no antigen. And what we're doing is we're going to add antibody against the antigen, right? So here's our patient sample. Here's a positive patient. Here's a negative patient. We're going to add antibody to it, okay? There's an immune complex forms. It does no here with the negative, right? Makes sense. But here it picks it up. The antibody picks it up. Then we're going to add antigen-coated particles, right? And those antigen-coated particles are going to pick up that there is no what? Antibody to bind. So there's no agglutination would be the end result, which is a positive test in this reaction. The negative patient who had no antigen, we added the antibody and we added the coated particles for testing, and there was agglutination. Okay, so the virus here, right, the patient with the virus actually competed with the antibody binding sites so that we couldn't get agglutination here when we look for it. Does that make sense? Hopefully. So it's I know it's against everything, but it's the way we do some testing. So actually, with this kind of testing, a negative result would be agglutination, where if there was no agglutination, we'd have a positive test. Okay. Here again, we can do that if we were looking for an antibody to the illness is a positive result versus no antibody here. We add the viral antigen to the sample. The antibody's there. We add the red cells to clump. They don't clump, so we get a positive test. Here we add the red cells. They do clump. That's a negative test. So all these illustrations are with our agglutination. And then we finish with this, putting it all on one slide to you, one slide, which is direct agglutination, indirect, reverse agglutination, hemoagglutination. And we will cover these each time we have one of these tests to perform. Okay, so don't think this is the last time you'll see it. But it is a principle that finishes up the chapter about how we can go about doing different methods of testing. All right, okay, so downstairs, 12:30, we'll do antigen testing with ELISA, and then we'll wrap that testing up for the week. And then on Friday, we'll move into chapter 12? Chapter 12 on Friday. I'm going to end Zoom.